Krishna. Hello everyone. This is Mrunmai, student of grade 10, and today I am here to explain you one of the wonderful topics from chemistry, that is indicators. Now you must be wondering that how are indicators useful in chemistry? We use indicators in our vehicles, but the indicators which you are thinking and which we use in chemistry are completely different. Now let us see what are exactly indicators in our chemistry. An indicator is a dye that changes color when it is added into acidic or a basic solution. Now one more question arises. What is the need to identify whether the solution is acidic in nature or what is the basic in nature? If solution is a basic in nature, what I want to do with that? But for our health, it is matter whether a solution we are consuming is acidic in nature or in a basic in nature. So now suppose I have given you 3 beakers. I know that in one beaker there is an acidic solution, in one beaker there is a basic solution, and in one beaker there is a neutral solution. But I don't know in which beaker which solution is there. And I have asked one of you to identify in which beaker acidic solution is there and in which beaker basic solution is there. Now we know that acids are sour in taste and bases are bitter in taste. Now my question will be asked that whether you will test the structure? Yes, definitely the answer will be no. Because we also know the property of acids and bases that they are corrosive in nature, which are harmful for us. Now, what can I do? I want that which one is acidic solution and which one is basic solution. I don't know anything else. So, now this wonderful indicator will help us to identify which one is acidic in nature and which one is basic in nature. So, indicators are divided into mainly 2 types, that are by changing color and by changing odor. By changing odor, indicators are also known as olfactory indicators, and by changing color, indicators are divided into 2 types, that are natural indicators and synthetic indicators. Now natural indicators, they are obtained from a natural thing. They may be from animals, unicellular, multicellular, or they may be from the plant. And synthetic indicators are indicators those we prepare in laboratories with the help of chemicals and many other things. So let us start with natural indicators. Moving towards the litmus, which we use. We have heard this litmus paper since many years. But what exactly this litmus is? It is obtained from the plant called lichen belonging to the division of group called Halophyta. Its original color is purple. Yes, right, purple. And it changes its color into red when it is added into the acidic solution. And it changes its color into blue when it is added into basic solution. Now, how exactly is this litmus paper made? Now, when we obtain the plant lichen, it is grind, solution is made, and then some amount of acid is added in it. Then it changes into red color, and then paper is dipped in, and then the paper is dried for some hours. Then we will get this red color paper. And if we will add some amount of base in that lichen solution, then we will get this blue color litmus paper. This was all about the litmus. Now let us move towards our second color changing indicator, that is turmeric. Yes, turmeric, which we use in our food. Haldi, turmeric, right. It is also a natural color changing indicator. Its natural color is yellow, and when it is added into the acidic solution, its color remains unchanged. But when it is added into the basic solution, its color changes into reddish brown color. Yes. So that's the reason why we observe that whenever a haldi stain remains on our white kurta, when we add the soap on it, it changes into the reddish brown color. Now let's move towards our next indicator, that is red cabbage indicator. Now when we will listen to the red cabbage indicator, we will little bit get confused that it's a red cabbage indicator, and in image we can see that it is a purple in color. Yes, but though its name is red cabbage, but it is in a purple color. So it is also a natural color changing indicator. Its original color is purple. Yes, this point we should always remember. the acidic solution, it changes into red color. And when it is added into the basic solution, it changes into the purple color. Moving towards the next indicator, which are synthetic indicators, means those are prepared in laboratory. First one is methyl orange. Methyl orange is a synthetic indicator whose natural color is orange, as the name indicates. When it is added into the acidic solution, it changes its color into red, and when it is added into the basic solution, it changes its color into yellow. Next one is the phenolphthalein, which is the most commonly used indicator, right? We have heard it many times. So its natural color is colorless. When it is added into acidic solution, the color remains colorless, and when it is added into the basic solution, it changes its color into pink color. Yes, right, pink color. Moving towards our next point, that is olfactory indicator. Olfactory indicators means indicators to changes their smell when they come in contact with acidic solution or basic solution. Commonly in our life, we see three main olfactory indicators, that are anila, onion, vanilla, and clove oil. So let us study in detail what exactly happened with these indicators. So onion, when onion comes in contact with acidic solution, its smell remains same as the smell of onion is in natural solution. So when it is come in the contact with basic solution, its smell will remain completely, means it will have no smell. So next, moving towards the vanilla. So when it is added into the acidic solution, its smell remains same. But when it is added into the basic solution, there is no smell. It will retain its smell completely. But in clove oil test, the case is completely different. When it is added to the acidic solution, the smell will remain same. But when it is added to the basic solution, the less smell will be observed. Means the complete smell is not vanished. Some amount of the smell is remained. So one trick we can make here, we can take onion from here and leaf from here. Anila we can make to remember that these two are same, but clove oil is different. So this was all from my side. I hope you all are clear with the topic indicators. Hare Krishna. Thank you.